In the chapter of Cell, the Unit of Life, today we are going to discuss about active and passive transport differentiation, osmosis and application of osmosis. I'm Dr. Hajra Yunus. So in this slide we will discuss about differentiate between active and passive transport. So in case of active and passive transport, the first point is movement of molecules against a concentration gradient, means from low to high concentration, and even in active transport molecules can move from towards the concentration gradient. So in active transport molecules can move from towards the concentration gradient, means from high to low concentration, or even against a concentration gradient, means from low to high concentration as mentioned in diagram definition. Whereas in case of passive transport, there is only movement of molecules towards the concentration gradient, means from high to low concentration. So if there is a movement against the concentration gradient, so something is required. So what is required? So in that case ATP is required. ATP is the energy. So energy is required for the movement of molecules against a concentration gradient, because if something wants to go against the concentration gradient, then something is required or some energy is required. In that case energy is ATP. So in active transport ATP is required, whereas in case of passive transport molecules are moving towards the concentration gradient, so it is a usual process, so nothing is unusual, so no ATP is required. So in that case ATP is not required. In case of, and third point is in case of active transport is affected by temperature and the poisonous substance, that is cyanide. It means if the temperature is fluctuating and there is a poisonous substance like cyanide is present, then the active transport is disturbed. Whereas in case of passive transport, because it is not affected by temperature or any other poisonous substance. So the examples of active transport are sodium and potassium pump, whereas in case of passive transport the examples are osmosis and diffusion. As you can see in this diagram, this is the diagram of active transport and this is the diagram of passive transport. So as you can see in the passive transport, here here the molecules are more, and here the higher concentration is high and here the concentration is low. So here what will happen, that the molecules will move from high concentration to low concentration, and here the process is towards the concentration gradient, so no energy is required because molecules are moving from high to low concentration. Whereas in case of active transport, here the active transport means the molecules move against a concentration gradient, means the molecules can move from low to high concentration. So as you can see here the concentration is high and here the concentration is low, so molecules will move from high concentration to low concentration, and after the process here the molecules are more, molecules have increased and here the molecules have decreased, but still here more molecules are required. So what will happen here, the ATP, that it is the form of energy, is required, and this ATP is used. By using this ATP or by the expenditure of ATP, here more molecules are taken in. So by using the ATP, the process of movement of molecules can be against the concentration gradient. So these are the differentiation between, or this is the differentiation between active and passive transport. In case of active transport, the molecules move against the concentration gradient, means molecules move from low to higher concentration with the expenditure of ATP, and whereas in case of passive transport molecules move towards the concentration gradient, means high to low concentration, and it is not unusual, so ATP is not required. So firstly we will talk about one of the examples of the passive transport, that is osmosis. Osmosis is the passive process, means in that passive process here the molecules move from high concentration to low concentration and ATP is not required. You will see in throughout the process of osmosis ATP is not used. So what is osmosis? Osmosis is the movement of solvent. In osmosis, solvent molecules are involved. For example, water. Solvent molecules will move from area of its high concentration to area of its lower concentration through semi-permeable membrane until it reaches in equilibrium. So in the osmosis, 3 points are very important. First thing is that in osmosis, solvent molecules are involved. Second thing is that in osmosis, semi-permeable membrane is essential. And third thing is that in osmosis, the process will take place until it reaches equilibrium. So this is the diagram of osmosis. In osmosis, this is the semi-permeable membrane, and this is the inside of the cell, and this is the outside of the cell. As you can see, as showed in the diagram, molecules will move from high concentration to low concentration. So as you can see here, the concentration is high and outside the cell concentration is low. So molecules will move from high concentration to low concentration, means from this direction to this direction, from inside the cell to outside the cell. Suppose this is the compartment A and this is the compartment B, and in the compartment A 500 water molecules are present, and in compartment B also 500 water molecules are present. So but out of these 500 water molecules, 100 salt molecules are also present. So 100 water molecules are occupied by salt molecules, so remaining water molecules are 400. And here the 200 salt molecules are present, so 200 salt molecules occupied the 200 water molecules, so remaining water molecules are 300. So as you can see here, the water molecules are more and here water molecules are less. So now the water molecule will move from high concentration to low concentration, means water molecule will move from compartment A to compartment B, or water molecules will move from inside of the cell to outside of the cell. So here the water molecules or solvent molecules are involved, and here the semi-permeable membrane is present, because in osmosis semi-permeable membrane is essential. And there is a third thing in the definition is until it reaches in equilibrium. Equilibrium means on the both sides the water molecules will be same. Like this is the diagram, and here the semi-permeable membrane is present, and this is the compartment A and this is the compartment B. So in case of equilibrium, here the 50 water molecules are free and here 50 water molecules are free. So in case of equilibrium, if in both compartments equal water molecules are present, so the state is called the equilibrium, and in equilibrium there is a no net exchange of water molecules. No net exchange of water molecules means if one molecule goes to the one compartment, so one molecule from the A compartment will go to the compartment B. So there is no net movement. You can say one molecule, if one molecule is going to A compartment, then one molecule is going from A to B compartment. So there is no net exchange or net movement of water molecules. So this state is called the equilibrium. So in osmosis, movement of water molecule or solvent molecules are required, and the always movement is from high concentration to low concentration. Semi-permeable membrane is essential, and the process of equilibrium osmosis takes place until it reaches equilibrium, and after equilibrium there is no net movement of water molecules. Now we will discuss about osmosis in animal and plant cell. So before going to discuss osmosis in plant and animal cell, we must know about three different terminologies like isotonic solution, hypertonic solution, and hypotonic solution. Means what are meant by these terminologies. In case of isotonic solution, the concentration of solute is the same inside and outside the cell, and water molecules move equally in the both direction. Like this is the for example, this is the beaker and here the cell is present, and this in the beaker solution is isotonic. If the solution is isotonic, it means that in the solution and inside the cell, the concentration of water molecules and the solute particles are same. So if there is concentration outside, if here in the solution and if in the cell, the concentration of water molecules are same, it means that there is now it is the equilibrium state. And now if there is an equilibrium state, then there is no net movement of water molecules. Means if one molecule is coming out, then one molecule is going in, so there is no net movement because one molecule is coming out, one molecule is going in, so still the state is equilibrium. So in isotonic, in the solution and inside the cell, the concentration of water molecules are same. So this is the isotonic solution. Next is hypertonic solution. In case of hypertonic solution, the solution has a higher solute concentration than the cell, and net movement of water molecule out of the cell causes it to shrink. And for example, and this is the beaker, and here the cell is present, and this is the hypertonic solution. Hypertonic solution means water is less and salts are more. So in the solution, water is less and salts are more. So in that case, water, it means water is less, and inside the cell water is more, so water will move from inside to outside. Whereas in case of hypotonic solution, the solution has a lower concentration, solute concentration than the cell, and the net movement of water molecule into the cell causes it to swell. So hypotonic solution is just the opposite of hypertonic solution. In hypertonic solution, hypotonic solution, water is more in the beaker and salts are less. So now in the solution, water molecules are more, so water will move from outside to inside. So these are the differences between the isotonic, hypertonic and hypotonic solution. Means in isotonic solution, the inside the cell and in the beaker, the solution in the beaker, they are in the equilibrium state and there is no net movement. Whereas in hypertonic solution, here the water in the solution, the water is less and the salts are more, so water will move from inside of the cell to outside, whereas hypotonic is just the opposite of hypertonic solution. Now in case of animal cell, for example, if the animal cell is in the uh, isotonic solution, so you can assume that this is the isotonic solution. Means the cell is placed inside the isotonic solution. As in the isotonic solution, because there is an equilibrium, so you can see there is the, here the water molecules move equally in the both direction. Means the one water molecule is coming out and one water molecule is going in. So this is the isotonic solution in case of animal cell. So there are two solutions, for example 0.9% NaCl solution and 5% glucose solution. These are the example of isotonic solution. If 0.9% NaCl solution is prepared or 5% glucose solution is prepared and the cell is placed in the solution, then there is a isotonic solution, or there is a isotonic condition. Whereas in case of hypertonic solution, so suppose this is the hypertonic solution, means here the water is less and salts are more. So here the if in the hypertonic solution water molecules are less and the salts are more, it means that the water molecules are more inside the cell. So as according to osmosis definition, water molecules will come out from high concentration to low concentration. It means here the water is more, so water will come out from inside to outside of the cell, and the process is called the exosmosis. Exosmosis means water is coming out, and water is coming out, and because of that the cell will get flaccid and will shrink. And you can see here the cell is you can see in the first diagram the cell is round and more or less round, whereas in the second diagram, because the water molecules have come out, because of that the shape has changed and the shape and the cell has shrinked. Whereas in case of the hypotonic solution, in here the water is more and inside water is less. So in that case water will move from high to low concentration, so water will move from outside to the inside, and because of that the cell will turgid. And if this entering of water molecules keeps continuing, then the cell will swell and will burst, and after some time this animal cell will burst. So these are the three conditions in case of osmosis in animal cell. The first is the isotonic. In isotonic, there is the cell is in the equilibrium state, so there is the equal movement of water molecules in both the direction. Whereas in case of hypertonic solution, outside water molecules are less and inside water molecules are more, so water will move from inside to the outside, and the process is the exosmosis, and because of exosmosis cell get flaccid and will shrink. And whereas in case of hypertonic solution the water molecules are going in, so this is the process of endosmosis. Endosmosis means the water molecules are going in, and the cell will swell, and if the process keeps continue, so after some time the cell will burst. Whereas in case of plant cell, so this is the isotonic condition, and the isotonic condition is similar to isotonic condition in animal cell. Whereas in case of hypertonic condition in plant cell, so there is a one difference between plant and animal cell. As you can see in the plant cell, there is a process of exosmosis, means the water molecules are coming out and the cell will get flaccid and cell will shrink. But and in case of animal cell, but in case of plant cell, as you can see, the water molecules are coming out, and but the cell will not get flaccid and cell will not shrink because of presence of cell wall. Because in case of plant cell wall is present, and the cell wall is responsible to provide turgidity and to shape not turgidity, but cell wall provides shape to cell. So because of presence of cell wall, even water molecules are coming out, or if water molecules have come out, the shape of cell has not changed. Whereas in case of animal cell wall is not present, so after process of exosmosis shape of cell wall has changed. So in case of hypotonic solution, as you can see in case of animal cell, there is a process of endosmosis, means the water molecules have taken in or have taken in. So because of that the cell gets flaccid, and after or not gets flaccid, cell will swell. And if this condition continues, then after some time the cell will burst, because animal cell does not have cell wall, whereas in case of plant cell wall is present, and because of that the cell will get turgid, but cell will not burst because of the presence of cell wall. So here are the applications of osmosis. So first application of osmosis is opening and closing of stomata, means how osmosis helps in opening and closing of stomata. As in this diagram, here you can see here the this diagram shows the stomata closing here, but this diagram shows the stomata opening. And how stomata gets opened? Because of the movement of water molecules, means outside the water molecules were more and inside water molecules were less, so water molecules move from outside to the inside, and now because of that the guard cells get turgid and the guard cells opened. Whereas another application of osmosis is the movement of mimosa pudica, which is commonly called touch me not plant, because when you touch the mimosa pudica, after some time you see that the plant so in case of mimosa pudica, as you touch the touch me not plant, the leaves show curling, because here the region is present, because this region is called the pulvinus, and as you touch the plant the stimulus is sent to the pulvinus region, and there will be the process of exosmosis, and in exosmosis water molecules come out, and in that manner, or in that case, as a response of touch, the release of touch me not plant show curling. Another example is the seed germination, means osmosis plays a very important role in seed germination. In seed germination, the first process is imbibition. Imbibition means the water molecules are adsorbed by the seed coat, means firstly the water molecules are adsorbed and resulting in the cracking of seed coat, and after that the water molecules are taken in by the embryo. So firstly there is a process of the imbibition, and then the water molecules are taken in. It means the water molecules were more outside and the inside the embryo water molecules were less, so the water molecules move from high concentration to low concentration. In that case, by absorbing the water, you can see the the plant has grown. So these are the three very important application of osmosis, like the opening and closing of stomata. Osmosis helps in opening and closing of stomata, movement of mimosa pudica, and even in the seed germination.